Our first speaker is Colin Phoon. I've known Colin for a long time because he comes regularly to our operating room to read echoes. And once we got our mouse, I actually have approached him and asked him to collaborate with us. And graciously had said yes and formed a very strong collaboration with Mindong. And he's going to present the results of that collaboration today.
I remember that day clearly. I was in the OR. Michael approached me, actually. Well, I'm thrilled to be here. I'm a newbie to... it's good to be in the OR, not for the patient. I'm thrilled to be here. I'm a newbie to the Barth conference, and I'm honored to be here, and I thank the organizers and the foundation for having me. We are going to talk about a mouse model, and actually the first talk by Dr. Jeffries was a perfect segue into this, as you'll see. You know, we're all interested in mitochondria because Barth syndrome is a mitochondrial disorder, and as it turns out, there's quite a bit of evidence emerging that mitochondrial disorders have some role in heart development. There are a number of different possible mechanisms, and I think people have tried to figure this out with Barth syndrome for some time, in particular because of myocardial patterning and noncompaction. We all learn in school that mitochondria are the powerhouses of the cell, but again, emerging data suggests that they are not just the powerhouses, and there are many, many different roles for mitochondria. I love this slide from Steve Claypool. He's got these little funny happy faces somewhere on his cardiolipin. Maybe it's on another slide. But as we know, tafazin encodes for the acyltransferase that will be involved in the maturation of the final cardiolipin species. And mitochondria really, and therefore also cardiolipin, are involved in not only bioenergetics, but apoptosis, calcium homeostasis, redox balance, which we'll talk something about, biosynthetic pathways, as it turns out, and important for heart development, transcriptional control, and cellular proliferation, as well as heme synthesis. This is coming out of Miriam's lab, and some immune responses. So this was one of the original seminal papers by Dr. Barth and his group, and it shows the clinical phenotype as well as some electron micrographs of cardiomyocytes and the abnormal mitochondria. And this is a picture similar to some of the stuff that we've seen of left ventricular noncompaction. So here's a still frame from Jeff Tobin's review more than a decade ago of the trabeculations that extend out into the left ventricular cavity. And as it turns out, noncompaction really is almost a form of arrested development. Trabeculations form from the myocardium very early in embryonic development. This is a necessary part of myocardial patterning, important in early heart function. And then these trabeculae, as it turns out, will eventually form the cardiac conduction system. And then the thicker part, the wall, will thicken up as the trabeculae sort of, I don't know if regress is the right word, but because they become less prominent. That whole process, as was alluded to, is really not well understood at all, but is important for normal heart function and normal myocardial development. In terms of models for Barth syndrome, we know that yeast, we've talked about a lot of yeast over the last day. Drosophila has not really been alluded to, but the Cincinnati group, Zaza Kucha, had published about a model of Drosophila. No, sorry, we've published Drosophila zebrafish from the Cincinnati group. Yeah, we've done the Drosophila. Sorry, Mindong. And zebrafish from the Cincinnati group a few years ago. Traditional mouse knockout genetics have been unsuccessful, but then the foundation commissioned for this knockdown strategy, which is summarized here. It's basically a doxycycline inducible system. It's a single mouse line, and it's a knockdown. The Florida and Cincinnati groups published this in 2011 and basically showed that the knockdown is successful. You can get it in adult mice of about 95% knockdown of tafazin. And interestingly, there is a cardiomyopathy. But it's a relatively mild cardiomyopathy, as shown by echocardiography and mouse MRI. And importantly, it develops in adulthood. They did not have a phenotype earlier in life. We played around with the mouse for a number of months. We were trying to just sort of get our feet wet, trying to figure it out. And I'll show you the data, and really it was very exciting. We published this just a couple of months ago. It's in the Journal of the American Heart Association. It's a brand new journal. You're not actually going to find it on PubMed, which is why I put it up here. We did actually publish it. It's on the Barth Syndrome Foundation website as well. But it's an open access journal, which means that you can access it for free, completely for free. Okay. So this was the day on the right-hand side. This was the eureka moment, one of those rare eureka moments that one may be fortunate to have. So it was November 2010. Again, we were putzing around trying to image some mice. And one of the things I like to do and have had an interest in a long time is imaging embryonic mice. We can image them and follow them in the litters from day to day throughout most of embryonic development. So this, and we had started the induction with doxycycline early in gestation, which is what you're supposed to do. This is a developmental problem. This is a gene defect you're born with. So this is an example of a normal wild-type embryo. This is the pregnant mother's abdomen. This is a uterine sac. This is a mouse embryo. The head is up here, and this is blood flow coming out of the heart. The spine is over here. This is what blood flow should look like in the dorsal aorta. In systole, during ventricular contraction, there's a nice antegrade flow. There is no reversal of flow during diastole. And then in the umbilical cord, there is nice arterial flow in the umbilical artery, and there's this undulating pattern in the umbilical vein. So we can look at all this. We can figure out some cardiovascular functional parameters. And then we came across this embryo, which grossly looked normal. This is the uterine sac. There's some amniotic fluid. There's a head here. There's a heart beating. And we saw this. So this is systolic flow in the dorsal aorta, but there was this extra kick, and there was also this other flow, which I'm not going to get into. But the extra kick was unusual, and I'd never seen anything like this in the, thousands of mouse embryos that I had imaged. And we kind of scratched our heads, and I said, Mindong was there, and I turned to Mindong, I said, Mindong, this is going to be one of our mutants. It'll be one of our tafazin heterozygotes. And it turned out to be right. This is 100% specific for the TAZ knockdown. It turns out this is the atrium kicking into the aorta. The pressures are low enough, and the ventricles, it turns out, is stiff enough, this is a cardiomyopathy, that the atrium can actually kick into the aorta, and it can kick all the way out into the umbilical artery. So this was a eureka moment for us. It was very, very exciting when we got the genotypes back. And in fact, as we looked further, again, here's normal mouse. This is at about 13 or 14 days gestation. This is a smaller one at about 12 days of gestation. That's nice and normal, very distinct structures, blood flow all the way around. And we started seeing dead mouse embryos as well. Prenatal lethality, which, as we heard from Colin Stewart, may well be a prominent feature that had not been appreciated until recently of families with Barth syndrome. And so here's a mouse that has not made it. There is no blood flow. I haven't shown any color, but there's no blood flow. But it's reasonably well formed. If you switch over to a short axis, there's a large amount of fluid, a pericardial effusion around the heart. This is one that's been dead for a little bit longer, and so it's smaller, it's getting resorbed, and there is some fluid around the heart. And this one's almost completely resorbed, so it's been dead for a little while. And we can almost time these things. This one's probably been dead for at least 2 days. So there's sort of a spectrum of when these embryos die. And if you look at the different gestational ages, more and more are dead later and later in gestation, and by newborn, many haven't made it, and then those that have made it actually won't make it beyond about the first few hours or first day of life. So there's substantial, almost universal, in fact, prenatal and perinatal lethality in this mouse. And we got to thinking, so why have we seen this when the other two groups didn't? We went back, we made some calls, we looked at the papers, and it turns out we ended up going by the company's protocol, Taconic Artemis. We were feeding our mice at least 3 to 10 times the amount of doxycycline that the other two groups were doing. They were going, I think, by their animal facility protocols. I also heard through the grapevine that their doxycycline was in the mouse chow, and the mice were smart enough to pick out the doxycycline and just eat the chow. So they might have been getting even less doxycycline. And so the knockdown may not have hit the critical period in development. That's what we speculate that we hit. So the next cool moment was when the histology came back a month or two later. This is a normal mouse heart, newborn. This is one of our tafazin knockdowns. Not only is the interventricular septum just sort of porous looking, it's not solid, but here is noncompaction. It was quite clear to us that this is noncompaction. The compacted zone is much thicker, I mean, it's all quantitatable, and then the trabecular region is much more prominent. This is in the newborn. This is at 13.5 days gestation. Same thing. Septum is not quite as well formed, and the trabeculae just look much more thick, spaghetti-like or worm-like. And so our TAZ mice in the end with the tafazin knockdown that we were inducing exhibit noncompaction. So this was this was exciting to see this in this model. As it turns out, systolic function, both in embryos and newborn mice, you can do all the numbers, you can crunch them, you can make measurements, and the systolic function was normal. We didn't see any evidence of dilated cardiomyopathy or ongoing heart failure, and in fact, that little blip, that atrial kick, suggested that much of this was diastolic dysfunction. The sudden death probably invoked arrhythmias, but we can't just hold the transducer there for 24 hours a day and look for arrhythmias, so we couldn't prove that necessarily. We had to, of course, prove that the cardiolipin biochemistry is altered. This is from Steve's paper. One of these is a happy face here. This one's a happy face cardiolipin. Most of them are abnormal and immature cardiolipin. And in fact, when we looked both at the embryonic hearts, this is just to show that the tafazin was knocked down at least 80% in mid-gestation. If you look at embryonic hearts, newborn hearts, there is a much higher monolysosyl cardiolipin to cardiolipin ratio, which is diagnostic, essentially, for this disease. If we look at the mitochondria, here are some normal mitochondria in wild-type mice, and here are our TAS knockdowns, very abnormal cristae. And in fact, even the relationship, one of the things we noted was the relationship to the myofibrils, which is emerging as being important, the relationship between mitochondria and the contractile fibers. That was disrupted in the TAS mice. This is in an adult from another publication, because it was a nice picture. This is our newborn wild-type mice. Here are nice elongated mitochondria closely associated with the myofibrillar units. The mitochondria in the TAS knockdown mice, in fact, were malaligned, misoriented when it came to the myofibrillar units. And we could measure a bunch of things like mitochondrial number, density, cristae density, alignment, and a lot of these were abnormal. The cristae density was relatively normal in the embryos, but became abnormal by the time they hit newborn. So a lot of electron micrographic evidence of mitochondrial problems, structural problems, which again we know are associated with abnormal cardiolipin. Some newer data, we look back just in the last month, and again here are some wild-type controls, and you can see, in addition to the mitochondria associated with the myofibrils, these dark areas are the Z-bands, which are part of the myofibrillar unit. And when we look back at the tafazin knockdown mice, we actually couldn't find as many Z-bands. So it looks like also the cardiomyocytes at the same ages were less well differentiated. As it turns out, so trabeculation in the mouse, the trabecular zone forms at around 9.5 days gestation, and it really starts to proliferate and becomes more prominent by 11, 12, and then it starts to sort of, the compact zone starts to thicken up, and compaction and then detrabeculation, I guess you could call it, is more or less complete by about 14, 15 days. What we did was we then timed the induction of the doxycycline. We decided to feed the doxycycline at various stages, and in fact, when we fed it at about 13 and 14 days gestation, we could not recapitulate the non-compaction phenotype, but when we fed it at 10.5 days, we could. And so it looks like the developmental window of what's required in terms of cardiolipin, normal cardiolipin, and the compaction process is a fairly narrow window of probably 2 or 3 days, which is along the lines of what one might expect knowing the compaction process. We looked very quickly as proof of principle at cardiomyocyte proliferation because other papers on other mouse models of left ventricular non-compaction have indicated a problem in proliferation. Some papers have indicated that the trabeculae are hyperproliferative. Others have indicated no change in proliferative rates. We took a look, and it looks like there may be differentially abnormal proliferation in the compact zone versus the trabecular zone. And when we did some counterstaining with a myosin-specific stain, phosphohistone 3 is a little different from Ki67, but it was just a sort of a confirmatory thing we did in the last month. And these are cardiomyocytes that are proliferating abnormally differently. We know also from trabecular basic studies on trabeculation and compaction that the whole cell cycling thing is very, very different in the two different layers. Trabecular layer actually exits cell cycle first, and then the compact zone continues to proliferate and thicken up. It looks like all of this is messed up in this mouse. The details are forthcoming, and that's part of our future work. one of the things we wanted to do was to fish around and see, well, what possible mechanisms might there be. So we took a look at some microarrays at E12.5, so sort of right in a critical period of trabeculation and compaction. We took a look at TAS knockdown mice and wild type that we only had duplicates. And surprisingly, we did not find a lot of energy processes downregulated. In fact, we found things like, I didn't know what to do with metal ion binding, but there's actually a lot of heme synthesis stuff and whatever else that doesn't show, that I didn't put in this table, that now I know from Miriam's group's work that might be, in fact, related and downstream of changes in cardiolipin, as well as steroid hormones. I was just having a conversation last night, which is very good. Zinc finger proteins and a lot of this other stuff, transcription, regulation of RNA processes, these are all transcriptional things. These may, in fact, have to do with cardiomyocyte proliferation and, therefore, heart tissue, heart muscle patterning, not energy processes. Other intriguing processes that look like they are changed as well: cell adhesion, cell migration, cell morphogenesis. These are all things that are going to go into tissue patterning as opposed to straightforward ATP synthesis and bioenergetic processes. Mindong will show some data on adult hearts that will show that it's a very different thing when you knock things down in the adult heart. So then we got interested in the, these are very recent data, and we got interested in the role of reactive oxygen species. Mitochondria, of course, generate reactive oxygen species, and there's a lot now written about oxidative stress, oxidative damage, and the roles of that in disease and aging. As it turns out, there's quite a bit of literature beginning to emerge on the mitochondria's role in development, embryonic development, and specifically on the role of reactive oxygen species, not as a damaging element, but actually as signaling molecules for proper development. And a lot of the work comes out of stem cell and very early embryonic research. This is actually an aging mouse, a progeria mouse. And then George Porter's lab up in Rochester just last year published this, and they basically manipulated the mitochondrial permeability transition pore and the levels of reactive oxygen species, and were able to show that you can change cardiomyocyte differentiation and all those myofibrillar units in development depending on the level of reactive oxygen species. So we thought this might be an interesting path to pursue as a mechanism for the myocardial patterning. When we stained, so we did a couple of preliminary things. These are fibroblasts from Barth syndrome patients. These are control patients. This was courtesy of Fred Voss. And when we stained the fibroblasts with mitosox red, in fact, mitochondrial ROS is increased in the Barth syndrome patient cells. These are mouse embryo-derived fibroblast cells. This is from courtesy of Zaza's group. And again, the knockout cells, the TAS knockout cells, have a much higher level of reactive oxygen species. When we looked at our TAS knockdown mouse cells at 12.5, this is DCF staining. This is just general reactive oxygen species. We have very limited numbers, but the littermate seem to show, littermate controls seem to show less ROS than the TAS knockdown. When we looked as far as 18.5 days, which is nearly the end of gestation, and this is mitosox red, which is specific for mitochondrial reactive oxygen species. Again, and I think I'm colorblind, so it's a little harder for me to see this, but I think there's clearly more red in these two as opposed to these two littermate controls. So again, more evidence suggesting that reactive oxygen species are increased when you have tafazin and cardiolipin deficiency. And that would make sense based on the literature. So one of the things we had to do, I mean, okay, so reactive oxygen species is increased, but does that actually have to do with anything? So the proof in the pudding would be if we try to reduce reactive oxygen species, can we rescue the phenotype? One of the things that had been published in the literature was the use of N-acetylcysteine. It's an FDA-approved drug to they actually use it in animal studies of aging diseases, but also there are a few reports of its use in embryonic development. It apparently crosses the placental circulation. It can get into the embryo. So based on that, and what N-acetylcysteine does, that it's most commonly used as a mucomist, although I think it's less used now for cystic fibrosis patients, loosens up the mucus and stuff. But it's also used in patients with renal failure. And before you give contrast for CT scans, which can damage the kidneys, this is supposed to help protect the kidneys as an antioxidant. And acetylcysteine is felt to be an antioxidant because it supplies, it's a precursor to cysteine, and it will maintain glutathione stores in this entire pathway. This is one of the pathways for antioxidant processes within the cell. So we fed our pregnant mice. We only had like 1 or 2 litters that we were able to do this so far. We fed pregnant mice N-acetylcysteine. And this is what we got. So this is the original, this is normal wild type, this is the non-compaction. This is a wild type newborn whose mother had been fed N-acetylcysteine, and this is a TAS knockdown fed N-acetylcysteine. And I think we haven't made the measurements on the entire litter, but I think that based on this and other figures, it's really quite suggestive that, in fact, the septum is much more normal, the compact zone is much more compacted, and there are fewer deep trabeculations as opposed to the non-treated pregnant mice. So this was very exciting to us, that the N-acetylcysteine might, in fact, be mitigating the non-compaction phenotype, and therefore supplying some, I think, reasonable evidence that the reactive oxygen species are underlying the entire non-compaction story in this model. We then further looked at some functional clustering of our microarrays, and in fact, 2 of the genes that the Finsterer paper, the review paper that Dr. Jeffries had put up in one of his slides, 2 of the genes that were listed actually fell out of our microarray in our TAS knockdown mouse. But then there are other genes that are related to compact zone and trabecular formation, transcriptional regulation, and then this particular pathway, PI3K-AKT, is a pathway that's involved in cellular proliferation, cardiomyocyte proliferation. So a lot of these genes fell out in our microarray analysis during functional annotation. So we do think, in fact, that something's going on with cell cycling. So this is sort of the model that we've developed, is that when you knock down TAS and alter your cardiolipin profiles, you have an increase in mitochondrial ROS. When you have an increase in the reactive oxygen species, it may have, in fact, differential effects and abnormal effects on the compact zone layer and the trabecular layer. The trabecular layer probably continues to proliferate, or actually in our mice it didn't, but because the compact zone may be hypoproliferative, you end up getting a thin compact zone while the trabecular continue to go on their merry way. In conclusion, the TAS knockdown mouse seems to be a pretty good model for human Barth syndrome, especially the fetal and infantile forms. We see hypertrabeculation non-compaction when we induce with our protocol. We see myocardial wall thinning. There's abnormal mitochondria, both structurally and functionally. And importantly, we think that reactive oxygen species are implicated. Mindong will show some data, in fact, that in adult hearts, when the heart is mature and you have knockdown and abnormal cardiolipin, it seems to be a very different cardiomyopathic process that's going on that may yield different targets for therapy. Mitochondria and heart development, in fact, are an emerging field, and we think that there's a really very probable role of mitochondrial reactive oxygen species, and not just bioenergetics. I think we really are talking about signaling molecules and cardiomyocyte differentiation and proliferation. There's a lot of other stuff that really can be explored. Calcium, cytoskeleton, migration, mitophagy, fission and fusion have been mentioned in the last couple of days as well. And I think looking at development might be one way to go in the production of this particular phenotype. I want to thank this entire team at NYU has been terrific since Michael approached me in the OR about 3.5 years ago. It's really been quite a joy to participate in this science. And really, I want to thank the Barth Syndrome Foundation for supporting this work especially. Thank you.
Colin, thank you very much. We have time for a few questions.
Obviously, I have to respond, as I said yesterday, that we see no clinical evidence of reactive oxygen species damage or involvement. And that's partly based on the fact that patients who clearly have that, who clearly respond to our antioxidant treatment, unlike the Barth patients, there's almost always a progressive component to the disease process, and that's the idea of the reaction to oxygen species causing damage. The other thing is that the choice of N-acetylcysteine was a little unfortunate because that's part of our amino acid treatment for Barth syndrome, and in fact, or cysteine is, and I've used N-acetylcysteine in place of cysteine as well. And in the first boy we treated with arginine, we couldn't get. He didn't really start getting better until I treated the cysteine, which was also low. It's just, I think it'd be worth repeating the experiment using a different kind of antioxidant because the cysteine could be acting in the way that the arginine does.
Yeah, so we're going to try a number of different genetic as well as pharmacologic approaches to reduce level of ROS. The other thing is that, clearly we haven't shown that treatment with NAC has reduced level of ROS in our embryos. The other thing is that, and I think we'll see from Mindong's data, we actually now believe that there are a couple of different distinct processes going on. So this involvement of reactive oxygen species will end up changing the myocardial template and producing the noncompaction. But whether that leads to ongoing damage in a more mature heart, in a functioning heart, in a postnatal heart, that's a bit more open to question, and they may not be as amenable to antioxidant therapy.
Okay, I have two questions. First one is, how strong is your evidence to prove that this ROS is generated by mitochondria? And second question is, did you check if the mitochondrial morphology is different between interfibrillar mitochondria and subsacramental mitochondria? Have you ever checked that?
Right. So the evidence, mitosox red, as I understand it, is mitochondrial specific. We do need more numbers. We need more litters. In terms of the readouts, like mitochondrial morphology and things like that, we really haven't done any of that. We looked in just a few mice, the levels of ROS. We have tried to treat just in a couple of litters with N-acetylcysteine. We got to fill in all the holes in between. Absolutely.
I think it's a little bit tricky to try to quantify the ROS by the method that you've shown. And if you want to be maybe a little bit more quantitative, you might try protein carbonylation.
Yes, so those are in the works as well.
We've shown that in the yeast has mutant, there's an increase in the protein carbonylation.
Yeah, we're going to start those experiments. One of the possibly tricky is that embryonic development is quite a short period of time, and we're not sure that we have enough time to be able to show differences in protein carbonylation or nitrosylation and other readouts. But yes, that's in the works.
So I have a question about this model is very useful in studying not only the Barth syndrome, which is an inborn error, but also to study what's really the physiological function of a test in adult mammal. So have you tried to induce test deficiency in, let's say, 2-month-old mice and follow up to see whether give them stress, let's say high-fat diet or whatever? Because what end up to kill most of our life, as you are the expert, is the heart attack, and that's beyond the Barth syndrome. So the question is, what is the test function, if any, in adult animal?
That will be the subject of Mindong's talk. So I won't give anything away. But yes, we have done that as well because we believe that there's a second distinct phase postnatally in which cardiolipin deficiency plays a role.
We have time for one quick question.
So just a quick comment on the ROS. First, so we've measured ROS release using the Amplex red assay in intact mitochondria from these mice, and it was actually lower. And I don't know that that's necessarily inconsistent with what you're seeing, as I understand it, mitosox is looking at the ROS inside the mitochondria. And so for people that are going to be looking at this further using a model such as the submitochondrial particles that allows you to turn the sort of the inner membranes inside out and look at ROS release from onto the inside, from complex 1, for instance, would be better. But anyway, I can talk more about that later. But my one quick question is whether you've looked at the dose response of doxycycline. I mean, this is very, very interesting, great work, and we don't see any real dysfunction in our adults that we've been studying until very far out, like we've seen in the papers that were published. Have you, been able to see an increased severity of this phenotype during embryonic development with different concentrations?
Yeah, that's a great question. We haven't had time to do that. If the other two groups were 3 to 10 times less, we probably would want to, half our dose or something as a starting point. But we were hoping to get around to that, but time has been short. Money is short.
Okay, thank you, Colin. We move on to